Okay, welcome back everybody. We are just going to finish up the splicing slides that we left off from last day, and then we'll start to move into translation today. So last day we had talked about the structure of the spliceosome and how we use all the components and the key features of the intron to carry out the splicing mechanism. And again, the splicing chemical reaction essentially consists of two transesterification reactions that will allow for the splicing of introns in eukaryotic mRNAs. Okay, so it's well known that mutations affecting splicing have been estimated to cause at least 15% of the genetic diseases that are known today. And I wanted to basically identify one of those diseases that you probably heard about. We're going to talk about thalassemia with this slide. So what is thalassemia? Basically, thalassemia is characterized by a reduced synthesis of the beta globin chain that makes up hemoglobin. So if you remember from your material from 210, hemoglobin is a tetrameric structure that consists of 2 alpha and 2 beta subunits. The clinical symptoms include anemia, and it is an autosomal recessive disease. Okay, so there is a splicing mutation that causes the thalassemia that leads to the decreased synthesis of this beta globin protein chain that makes up hemoglobin. And we're going to actually demonstrate how this mutation occurs. So if we focus in on this transcript right here, which is the normal pre-mRNA for the beta chain. So we're just going to write beta chain here. And we've just depicted the key area where this mutation occurs. So if you focus on here, this is an intron. And of course, we can have exon here and another exon here. And of course, this region has to be spliced out to generate the mature transcript. So spliced out. And this is what occurs in the normal pre-mRNA processing. In the case of thalassemia, within the intron, there is actually a key A nucleotide that gets mutated to a G. And we can see that here. Okay. We notice that in our normal intron, we have the invariant GU at the 5' splice site, and also our AG at the 3' splice site that allows for the spliceosome to actually assemble and carry out those two transesterification reactions. However, when we get this mutation happening, so going from the A to the G, well, what actually happens is that we introduce a new 5' splice site. So we can see it here, at GU. So not only do we have one here, we now have a second one within the intron. And this, of course, results in the possibility of U1, which recognizes this 5' splice site, either starting to assemble here 50% of the time, or assembling here 50% of the time. Okay? Now, when the assembly occurs at this new 5' splice site for the spliceosome, you realize that the only part of the intron that gets cleaved out is this region. So only this portion is cleaved. And we're still left with this component of the intron that remains. Serendipitously, unfortunately, as you move along this intron, there's actually a stop codon right before that new 5' splice site. So rather than having the ribosome translate the entire transcript, once it reaches this critical point, the stop codon, it will actually truncate the transcript when it's being translated. So you get truncated translation because this remaining part of the intron has now become incorporated into the mature mRNA. It is abnormal, and once the ribosome reaches this point, it stops, and then you get an abnormal protein, which essentially then becomes degraded. Now, if the assembly again is over here at the correct site, you actually get a normal transcript. Okay? So again, just to reiterate, the A to G mutation within that first intron of the gene for the human beta hemoglobin protein creates a new 5' splice site, so a new GU. And again, it's recognized by this area right here. Both the 5' splice sites are recognized by the U1. So splicing sometimes may create a normal mature mRNA, and 50% of the time an abnormal mature mRNA that contains an intron sequence. The normal mature mRNA is then translated into the correct normal beta globulin protein because it includes this one over here includes an intron sequence. The abnormal immature mRNA with that stop codon is actually degraded. Okay, so that's how mutations that affect splicing can actually introduce genetic diseases. And the case we discussed here is for thalassemia. Okay, so let's talk about tissue-specific splicing and processing. Okay, and we're also going to focus on two different proteins as well in this slide. And one of them is calcitonin, which is depicted down here. Calcitonin is responsible for regulating, just write this here, regulating calcium and phosphorus metabolism. And over on this side, we have another protein, CGRP, which is the calcitonin gene-related protein. So calcitonin gene-related protein. And again, this one is responsible for vasodilation. Vasodilation. Both of these proteins are actually created from a single primary transcript, which is depicted up here. The interesting feature about this primary transcript is that you actually have two polyadenylation sites in the transcript. So number 1 and number 2. Okay, so the fact that we have not only these two different polyadenylation sites, we also have alternative splicing of this transcript occurring. So alternative splicing. Okay, so alternative splicing really does generate diversity, and we'll see from the generation of calcitonin and CGRP that these are two different hormones that are produced from a single calcitonin CGRP pre-mRNA. Okay, and it is tissue specific. So depending on where the transcript finds itself, whether it's in the thyroid, so we have two different tissues here, or in the brain, we can get two different proteins being generated. Okay, so the different combinations of the exons within this primary transcript from the same gene may be spliced into a mature mRNA, producing distinct forms of a protein that's specific for tissues and for signaling pathways. Okay, so it's just another way of providing a powerful mechanism for expanding the versatility of these genomic sequences. Okay, so if we look at this transcript, and if it exists in the thyroid tissue, okay, we do get inclusion of exon number 4, okay, and recognition of that first polyadenylation site, depending on the factors that are there, and we get a truncated transcript that is eventually processed. So we get our 7-methyl-G cap that's added on. So we've got 7-methyl-G and our poly-A tail that's been added on. And then again, our introns are spliced out to get the mature mRNA. This will then lead to the ribosome translating this protein, this transcript into the calcitonin protein. Okay. In neuronal cells, however, we have alternative splicing of exon 4. So alternative splicing and recognition of the second polyadenylation site in this transcript. Okay, so exclusion of exon 4, and again, as we add on our 7-methyl-G cap and our poly-A tail, and we get splicing out of the introns plus exon number 4, we get a mature mRNA that is lacking exon 4. So lacking exon 4 and the brain tissue, and then hence, once the ribosome translates it, we obtain the calcitonin gene-related protein. Okay, so the key thing to take away from this is that the primary transcript has 2 polyadenylation sites, so number 1 and number 2, and one will predominate in the brain and the other in the thyroid. And in the brain, again, splicing will eliminate exon 4, which is sometimes also referred to as the calcitonin exon because it's found only in calcitonin. And in the thyroid, that calcitonin exon is actually retained. Okay, so and then the resulting peptides are then processed further to yield their final products. Okay, so again, let's just do a quick reminder about the splicing processes that we have for tRNAs and rRNAs. We went through this last day. Again, we have a couple of 5 steps. We have the cleavage of the leader region. We have the cleavage of the trailer region. Okay, we have the addition of the CCA. And again, there's an enzyme that will add that using 2 CTPs plus 1 ATP to do so. And again, this is where we're going to attach our amino acid, as we'll see in the translation lecture. We get base modifications occurring in the D loop, the T size C loop. So the D loop. Again, we have some methylations here. And then we have that splicing in the eukaryotic cell of this component here. So you get splicing as well. Okay, so that's so these base modifications are number 4, and then the splicing is the 5. So 5 processes that are occurring to get our mature tRNA. And you can see here that we have a superscript of the amino acid tyrosine. This denotes or specifies the addition of tyrosine to this amino acid here arm. So we would add a tyrosine based on the anticodon sequence that we see at the bottom of the tRNA. Okay? And we'll see how these tRNAs assist in protein synthesis in the translation section of the course. Okay, so another reminder, we have all these base modifications, and again, I definitely want you to focus on these. They are important. We've been through some of these in lecture 3, or in the first half of the course. I think it was lecture 3, if I'm mistaken. Again, we have 4-thiouridine, where we have a sulfur group attached in that position number 4. instead of an oxygen. Inosine. Inosine will become important when we start talking about protein synthesis. It's found in a high concentration in the anticodon region of tRNAs. So anticodon. We have methylation, and again, this is 1-methylguanosine on position number 1. We have another interesting base modification, N6, isopentyladenosine, sometimes referred to as I6A, and the N here actually refers that we have this isopentyl group, which is depicted in red on that nitrogen, position number 6, and then we have it on the base adenosine. Okay, ribothamidylate. Again, we have uracil with that methyl group, and our sugar is a ribose, so it's called ribothamidine. Pseudouridine, and again, we have the shift from the ribose link from position number 1 to number 5, and then dihydrouridine, again, where we lose that double bond, and we have the 2 hydrogens that are attached to dihydrouridine. So that's found in the D loop. Okay, so at this point, I really want to introduce you all into some of the self-splicing introns that were first discovered in the early 80s, and compare these self-splicing introns to what we've actually just discussed, the structure and function of the spliceosome in characterizing and splicing out nuclear mRNA transcripts. Oops, that should say mRNA. Okay. So we're going to look at a comparison of these signaling splicing pathways. And again, if you notice on these slides, the exons that are being joined are shown basically in blue and yellow. And the attacking unit is actually shown in green. So we have the unit here, and then we have another one here, and then of course what we talked about, the key A found in the branch site in the intron that is recognized by U2 as part of the spliceosome. Okay, the catalytic site in these introns is also depicted in red. We can see that for these group 1 and group 2, these self-splicing introns, that it's actually a cofactor for group 1. So this is a cofactor that actually comes in, a G, and this G can be GMP, GDP, or GTP. Whereas for group 2, the catalytic site is actually part of the intron. So it's part of intron. And then also in the spliceosome, the key A is also part of the intron. What you also notice, key difference between group 1, 2, and the spliceosome we talked about last day, is that for group 2 in the spliceosome, we are using the 2' hydroxyl to carry out the first transesterification reaction, whereas group 1 will actually use the 3' hydroxyl from the cofactor that comes in to carry out this transesterification reaction. Okay, and these self-splicing introns were actually identified from mRNA precursors in mitochondria of yeast and fungi that undergo self-splicing. So the self-splicing reaction can actually be classified according to the nature of the unit that it attacks upstream of the splice site. So for group 1, as we noted, it's mediated by guanosine cofactor. Group 2, the attacking moiety is the 2' hydroxyl from a specific adenylate that's found in the intron. And again, for the spliceosome, we still have that 2' hydroxyl from a particular adenylate that's found in intron at the branch site. Okay, so the group 1 and the group 2 self-splicing introns actually kind of resemble the spliceosome catalysis in two respects. So in the initial steps, right, a ribose OH attacks the 5' splice site, and we'll see that as depicted by this arrow here and this arrow as well. And then the newly formed 3' terminus of the upstream exon then attacks, so if we go step 2 down here, so this newly formed 3' OH hydroxyl will then attack the 3' end of the splice site of the intron of splice site. And again, down here, the 3' splice site. Okay, and to form that phosphodiester bond with the downstream exon, so that we join the blue with the yellow. And again, the group 2, the same along with the spliceosome. And then both reactions are transesterification reactions in which the phosphate moieties on each of the splice sites are retained in the products. Okay, so the group 2 is more like the spliceosome, and the attack is carried out by the intron itself using that 2' hydroxyl A rather than an external G cofactor. All right, so let's just go back in history and actually look at some of the experimental data that led to the discovery of these self-splicing introns. And most of this work was carried out by Thomas Cech, who was from the University of Colorado, and he characterized the group 1 introns, and also by Sid Altman, who was from Yale, and also characterized the RNA activity in RNase P, which is what we've already noted, a tRNA processing factor. And this figure over here is actually the group 1 self-splicing process, which we'll delve into in more detail in the upcoming slides. And as you notice, it's the group 1 because we have that G cofactor that's essential for the first transesterification reaction to occur. Okay, so some of the experimental data we're going to show is from both Thomas Cech and Sid Altman's groups, and both of them were awarded the Nobel Prize for their work with these self-splicing introns, which they essentially called ribozymes. Okay, so we can actually define different classes of these ribozymes, and we've talked about the group 1 and the group 2 introns very briefly. So what is a ribozyme? If you can think back to the first lecture of the course, when we defined the different types of RNA, we did bring up the definition of ribosomes. So basically, they are RNA components that will use RNA molecules or themselves as substrates for catalysis. Okay, so if we have a ribozyme, the group 1 and the group 2, and of course the spliceosome will fall under these the category or definition of a ribosome. So again, for the group 1, we're going to actually focus on a specific species, a eukaryotic cell called the tetrahymena, which a lot of this experimental data is revolved around. And again, it's part of the group 1, and the group 1 introns basically are found in eukaryotic genomes in the rRNAs in the tRNA transcripts. Okay, and these ones, of course, key thing, you require that G cofactor, which uses the 3' hydroxyl as that nucleophile. Oops, 3' hydroxyl as that nucleophile to carry out catalysis. Okay, so they are self-splicing. They don't need ATP. And again, it was first discovered in 1982 using the eukaryotic cell tetrahymena, so 1982. 82. All right. The group 2 introns are mostly discovered in mitochondrial genomes and the mRNA that we see in these genomes. And of course, the key difference here, instead of using that 3' hydroxyl from the G cofactor, we use that 2' hydroxyl from the adenylate that's found within the intron. Okay. Other types of classes of ribozymes include the viral RNAs, which we don't have time for in this course, but are a very interesting group because they're really just small RNAs that are associated with RNA viruses. Okay, and we can define a couple of them here: the hammerhead ribozyme, the hairpin ribozyme, and then the hepatitis delta virus ribozyme. And again, these will actually use different mechanisms to carry out these transesterification reactions. So I definitely encourage you to look up a couple of these viral RNAs to see their how they actually contribute to ribosome activity. RNase P, again, that was described by Sid Altman for tRNA processing in bacteria. And again, it produces that 5' phosphate in the 3' hydroxyl groups at the site of cleavage of the tRNAs. Okay, tRNAs. Again, I put some question marks here beside the spliceosome and the ribosome. And again, key thing here is that both of these require proteins as well. So they're not entirely RNA that carry out these catalytic mechanisms, but they also use proteins. So I'll really leave it up to you to decide as whether they fall under the definition of a ribozyme. I mean, according to myself, I believe they are ribozymes. They carry out these catalytic mechanisms, and they are RNA components in addition to proteins that actually carry out these processes. Okay, so let's look at the actual mechanism, splicing mechanism, that's carried out by these group 1 introns. And again, the self-splicing, the versatility of it, first became clear from the observations of this processing of rRNA. So when we talk about the tetrahymena, we were using an rRNA transcript from the experimental data. And again, it's associated protozoan single cell eukaryotic cell tetrahymena. Okay, so the catalytic mechanism, much like the spliceosome, is a series of transesterification reactions. So we can see we have our upstream exon in blue that we need to join with our downstream exon in yellow, and the intron lies in between at the 3' end of the exon number 1, and then also here at the 5' end of the exon number 2. The splice sites basically are aligned when base pairing occurs between the internal guide sequence, so the IGS, which is this region right here within the intron, so the internal guide sequence, or the IGS, so IGS, base pairs with the 3' region of exon number 1. Okay, so you get the base pairing that occurs with the internal guide sequence in the intron and the 5' exon and its terminal region right here. Okay. We then next bring in the G cofactor into basically an active site. So we'll look at the active site in a few minutes. Okay, and this helps align the 3' hydroxyl for nucleophilic attack at the phosphate at this 5' splice site. Okay. The IGS then holds the downstream, so we get cleavage, and so we're going to go up here. We're going to get cleavage at that 3' end of exon number 1, and incorporation of the G into the 5' end of the intron. Okay, so that's the key thing with the group 1 self-splicing introns. You get incorporation of the guanosine cofactor. The IGS then holds the downstream exon, so we have that free 3' hydroxyl for attack by the newly formed 3' hydroxyl to the upstream portion right here of the exon, so that we can join exon number 1, which is in blue, with exon number 2. We get the second transesterification reaction. We get joining of the 2 exons down here, and release of the intron. So release of intron. Okay, so the first step in this self-splicing reaction is basically alignment of the internal guide sequence or base pairing with the upstream exon. The incoming guanosine cofactor is then brought into the active site, which aligns the 3' hydroxyl of the guanosine cofactor at the 5' splice site. We get the first transesterification reaction. Here's our first. And this leads to incorporation of the guanosine cofactor into the 5' end of the intron. And then the IGS will then hold the downstream exon in position for attack at the 5' end of exon number 2. And we get the second transesterification reaction, so step number 3. And then finally, release of the intron, and our finally exon number 1 and exon number 2 that are completely joined. Okay. And this is just more a schematic at the molecular level, so we can actually visualize the G cofactor that comes in once we've base paired the IGS with the 3' end of exon number 1. And you can see here it's that 3' hydroxyl that carries out nucleophilic attack onto the phosphate. So nucleophilic attack. So we break a phosphodiester link, and then we create a new one down here. Okay. And, oh, sorry, this is the second transesterification reaction. Sorry. So we actually break a bond here and create a new one when we incorporate the G into that 5' end of the intron. And then finally, for the second transesterification reaction, we get nucleophilic attack between that GU of the 3' hydroxyl of the first exon. So that we attack here, we break a phosphodiester bond, and then we create a new one when we join exon number 1 with exon number 2, and release the free lariat intron component. Okay. All right. So in that group 1 intron and tetrahymena, what Thomas Cech actually discovered was that the intron that was actually released was 414 nucleotides in length. Okay, so again, another schematic about the reaction that occurs in tetrahymena, and this is in an rRNA transcript. Okay, there's a particular intron that will be removed using that guanosine cofactor or 3' hydroxyl, we get the first transesterification reaction and incorporation of the G into the 5' end of the intron, and then that free 3' hydroxyl from the 3' end of the exon number 1 will nucleophilically attack that second splice site, so we can get the second transesterification reaction, joining of the two exons together and release of the intron, which is 414 nucleotides in length. Okay, so this intron, again, becomes important because we'll see that it actually does something else to itself. So it is actually characterized as a ribozyme on its own once it's been cleaved from the primary transcript. And what does it actually do? This slide will depict how the intron itself, so once we've created that intron, actually splices itself twice, so 2 more times, so 2 more splicing reactions, okay, to produce a linear RNA that has or becomes L19, okay? And basically, this initial intron of 414 nucleotides will circularize after it's carried out the transesterification reaction and lose 16 or 15 nucleotides. Once it circularizes, it opens up again, carries out another transesterification reaction and loses 4 more nucleotides, so minus 4 more nucleotides, to generate the L19. And it's called L19. The L stands for loss, and the 19 comes from the 4 and the subsequent 15 nucleotides that are lost during these 2 transesterification reactions. Okay? And again, what we'll see in a couple of minutes is that this L19 is actually still catalytically active. So it carries out a specific reaction itself, and we'll see that in a couple of slides. Okay, so just a bit about the structure of the self-splicing RNAs. They're not just very flat 2D structures. They actually have very complex 3D structures. And this is, again, from Tetrahymena. All right. And that self-splicing intron has a very complex folding pattern. And as you can see, of helices and loops. All right. And what's depicted here by the different colors are the bases. So green will denote A, yellow C, purple G, and orange are the U's. Okay. Much of the structure is double helical, with stems and loops, and a pocket for the binding of the guanosine cofactor. There's lots of coordination with magnesium ions, not only in the structure, but also in the active site as well. So we're going to have lots of magnesium ions that are associated with this structure. Okay, and again, the 3D structure is important for the function. And we know this because if you actually inactivate it by heating, so you lose the 3D structure, it cannot carry out catalysis, by heating. So, and again, has a specific TM. If you denature the structure by heating it, you lose the catalytic activity of the self-splicing intron. Also, if you add in, so that's number 1, and number 2, denaturing agents, which disrupt the base pairing. So disrupt base pairing, the extensive base pairing that's occurring in this 3D structure, again, you get a decrease in catalysis or these transesterification reactions. And also if you mutate, studies have also shown if you mutate specific bases, so essential nucleotides, these will all decrease catalytic activity. Okay, so the 3D structure is highly critical for the self-splicing intron to carry out its particular function in the pre-rRNA transcript in tetrahymena. Okay, so let's really focus on some of the data, experimental data that Czech was also able to publish and demonstrate the self-splicing ability of these group 1 introns. And this group 1 intron that they identified was actually called the IVS, so the intervening sequence that was excised in the rRNA, not mRNA, of tetrahymena, and that resulted in the generation of the 17S, the 5.8S, and the 26S, which all make up the ribosome. And remember, we talked about how all of these components are typically generated in a single pre-rRNA strand, so that one copy of each is created to help synthesize or form the ribosome to carry out translation. So there was a particular IVS, or intervening sequence, or intron, that had to be removed to generate the full-length 26S transcript that's essential for the ribosome. And what Czech actually did was, as depicted from this plot from one of his experiments, he looked at the ability of this intervening sequence to either circularize or linearize after self-splicing. Okay, and he also tried to figure out whether this process was protein dependent or protein independent. Okay, so if we look at this blot, basically what we have in this lane S is the pre-rRNA transcript. So basically the entire transcript that we've denoted over here on the blot. So we can see it has a very large molecular weight. He also isolated in lane M both linear and circular IVS. And again, these are both controls, so they can be used as a comparison for your experimental procedure. And then, as we can see, if you should probably draw a little band here, both the circular and the linear forms after transesterification have occurred are denoted by these two bands at the bottom. Okay, so when we cut out that 414 nucleotide intron, it can either be in the circular form or the linear form, and they're denoted here on the blot in this region. Okay, so the first thing he did was actually look at the role of magnesium in the catalytic process for this IVS, and in these two lanes that we denote underneath the umbrella of NT, so again, NT is no treatment. We can see that only in the presence of magnesium do we actually get the circular and linear forms of the IVS appearing on the blot. So that was very critical, that established the role of magnesium in the IVS and being able to carry out the catalytic function of creating both the circular and linear form of the intron. So the self-splicing of it from that pre-rRNA transcript. In the absence of magnesium, nothing was excised. So the IVS was not removed. Okay, so that was key. So the one thing that did come out of the experiments, you actually need magnesium to carry out catalysis. Okay, we already know it's essential for its structure, so that is one rule. Another question which he did later on was ask whether it was essential for catalysis, so like in the active site. And we'll look at those experiments in just a few minutes. And then in the lanes beside the non-treated, he incorporated 2 different proteases. Now you know that proteases will cleave up proteins, and you can see in the presence of magnesium, again, we both get the circular and linear forms of the IVS appearing. So this basically established that the IVS did not need proteins to carry out its self-splicing mechanism. So catalytic processes were protein independent. Okay, and again, as controls with no magnesium present, there was no circular or linear form appearing on the gel. Okay, so some experimental evidence that first established that magnesium is critical for the IVS to self-splice. And again, we see that with the appearance of the circular and linear forms when magnesium is incorporated into the experimental condition. And then finally, that the IVS does this self-splicing independent of proteins, because in the presence and absence of proteases, which degrade proteins, we still are able to see the circular and linear forms appear on the gel. Okay, so again, the magnesium we know is highly critical in establishing this 3D structure that we talked about with the group 1 introns. The group 1 rRNA has an obligatory divalent cation requirement, and common divalent ions that it can use are, of course, magnesium, manganese, can also substitute for maintaining the 3D structure. Okay, and again, if we're looking at the molecular level, this is just a region of the double helical loops and turns that we actually see in the IVS, and more molecularly, we can see the magnesium ions being depicted here as these circular spheres. And if you remember back, we talked about how magnesium can promote the global folding of RNA. So the global folding of RNA, and it does so by coordinating with phosphates. If you can remember back when we talked about this, magnesium can also coordinate with water, and the water can then hydrogen bond to bases, and then finally magnesium can also coordinate with the water to the 2' hydroxyl, all leading to stabilization of the 3D structure. All right, so another key thing that they wanted to address in their experiments was to actually look at the guanosine binding site, and to understand how the guanosine cofactor that comes into these binding sites in the self-splicing group 1 introns, how does it actually facilitate that first transesterification reaction? And if we look at the figure over here on the left, we actually have a space-filling model of one of the regions that's denoted over here in blue, called P7. Okay, so P7. So this is the paired region number 7. So paired region number 7, which makes up the active site. And you can see very vaguely down here, this is our G cofactor that's actually come in to the active site in that 3D self-splicing intron. Okay, so when that G cofactor actually comes in, it forms a triple base pair between a GC pair, which are these ones here, that is found in that paired region number 7. Okay, and we know this because site-directed mutagenesis of this site, and specifically the CG or Watson-Crick base pair, has destroyed splicing activity. Okay, and again, the binding of the G cofactor to tetrahymena is saturable. Okay, and again, you can see that we get a triple base pair that is critical, that forms between this Watson-Crick base pair in the active site or the binding site of the paired region number 7 and the intron, the group 1 intron. Okay, and we're going to look at some experiments that actually showed the site-directed mutagenesis of this site and the catalytic activity that's associated with these mutations. So basically we have, again, the normal triple base pair. So normal triple base pair that's occurring in that paired region number 7 between our normal Watson-Crick and then our G cofactor that comes in. All right. So one of the first experiments they did was they actually took this G and turned it into an A, so they mutated it. And when they did that, they found that there was no activity, no catalytic activity, no splicing mechanism occurring. The next thing they did was they took the C and mutated it to U. So you still had, after this mutation, a normal Watson-Crick base pair, an A with a U, but in the presence of that G cofactor, again, there was no activity. Okay, so site-directed mutation of the G, the 264 to an A, destroyed splicing activity, and activity was not restored by the compensatory C to U change, so that we had the normal Watson-Crick base pair of AU at that position and at 311. So this technically just destroyed the catalytic activity. However, the critical specificity change or experiment that they conducted was actually using that mutated AU Watson-Crick base pair. So mutated double mutant that essentially had substantial activity restored in the first step of the self-splicing mechanism when they introduce the cofactor 2-aminopurine, which is denoted over here. So 2AP is 2-aminopurine. Okay, so when they had this double mutant, in the presence of the G cofactor, they got no activity. However, when they supplied it with the cofactor 2AP, they were able to restore substantial activity to the intron and its self-splicing mechanism. And the specificity change that occurs can be best understood in terms of the nucleosides binding in the major groove of the P7 region. Okay, so let's look at the data from this double mutant experiment. Again, I've listed the paper below, which helped to analyze the guanosine binding site of the tetrahybrid ribozyme. Again, some of the key things that we're looking at here are Km values. If you think back to 210 in your enzyme section, Km is a measure of affinity for a particular enzyme. V is for velocity. If you forgot these terms, please review them. And then V over M basically is just a ratio of the two to define the catalytic efficiency of the reaction that's occurring. Okay, and what we have in this table, if we were to create a table, is basically the wild type, so the normal Western GC base pair, and then the mutant that was created using AU as the Watson-Crick base pair. And two conditions were carried out in these experiments. One where the wild type was supplied as guanosine cofactor, and we can look at the values for the Km and the velocity. So we can see that the Km is very low, which means it has a high affinity for the guanosine cofactor. And then when we look at the catalytic efficiency of the wild type in the presence of the G cofactor, it's very, very high. So extensive catalytic activity. However, again, when the normal Watson-Crick base pair GC was exposed to 2-aminopurine, the catalytic efficiency dropped dramatically. Using the mutant, the double mutant, AU, in the presence of guanosine cofactor, we saw that catalytic efficiency decreased tremendously. However, if 2-aminopurine was supplied to the double mutant, you were able to restore activity. So you can look at the two different combinations of the wild type with the mutant. This can best be explained by the chemistry and why the AU double mutant worked in the presence of AP. And again, it has to do with the terminology that the two, the wild type and the mutant, were isosteric in nature. Okay, so what does isosteric mean? So basically they have the same number of valence electrons in the same configuration, but differing in the kinds and numbers of atoms. Okay, so basically the similar electrical arrangement in these two, in both the wild type and the mutant, but again, this is carried out through different and differing in the number and kinds of atoms that are used. Okay, so having the same number of valence electrons in the same configuration. And this is why the double mutant in the presence of 2-aminopurine was actually able to restore catalysis to that particular intron. All right, so let's get back to L19. And if you remember, I briefly stated that this is also catalytically active. Okay, so catalytically active. And I just want to briefly talk about what it actually is able to do. So it's defined as a catalyst because the L19 actually promotes nucleotidal transfer reactions. So nucleotidal transfer reactions. And this nucleotidal transfer reaction is basically where some oligonucleotides are lengthened at the expense of others. And the best substrates for are oligonucleotides that are synthetically approximately C5 in length, and that's denoted here. So we can have an oligonucleotide that's made up of C's and approximately 5 nucleotides in length. So if this is our L19 intron, how this process works is that this oligonucleotide can come in and base pair with the 5' end of the intron, okay? And this 5' end is very G-rich, hence why it really likes to use synthetic C oligonucleotides, okay? And as we follow the process, when this base pairs, it aligns that 3' end hydroxyl at the first C in that oligonucleotide to carry out a transesterification reaction. So we get a first transesterification reaction, which then results in incorporation of one of the C's into that 3' end of the intron, okay, and release of the 5 C oligo, which has now been converted to a 4 C oligo. Okay, it will then take another C5 base pair with that 5' end, that G-rich region, and the 3' hydroxyl on that new oligo C will then attack the 3' end of the intron and incorporate that C that's in the intron into the original oligo 5C and create one that is now in length with 6 C's and having released the oligo with 4 C's. Okay, the intron basically acts as a catalyst. It follows Michaelis-Menten kinetics, and it's very specific for this synthetic C5 oligomeric structure. And again, it can keep doing this over and over and over again. Okay, so it does this nucleotidyl transfer reaction so that it lengthens one oligonucleotide in length and then decreases another, so at the expense of another. Okay, so again, 2 transesterification reactions. Here's the second one down here, second transesterification. Again, lengthening one oligonucleotide at the expense of another. Okay, so basically the last thing that left to be characterized for these group 1 introns was whether, it had already been shown that magnesium was required for the 3D structure, but whether the magnesium was actually required in the active site. And again, if we look into the active site, if we just imagine that this is the active site where we have the guanosine cofactor, which is depicted down here, coming in, or cofactor, which is also coordinated to magnesium ions. And again, here would be our paired regions between number 9 and number 2, and where our first transesterification reaction needs to occur. We have our 5' end up here, followed by a 3' attacking that phosphate using the 3' hydroxyl that's come in from the guanosine cofactor. And hypothetically, if we had magnesium as well, that's also coordinated within the active site to help facilitate this reaction. We would anticipate that the magnesium helps orient the substrate so that covalent catalysis and metal ion catalysis can occur. And these typically are strategies that are used by protein enzymes, which we can hypothesize and translate to RNA or ribozymes, so RNA components that actually carry out catalysis. And again, the magnesium ions, we can potentially hypothesize that they are directly involved in facilitating this catalysis. Hopefully, as depicted here, by stabilizing the transition state. Stabilizing the transition state during the catalytic reaction. And the critical experiments that were actually carried out by Czech to identify the role of magnesium ions in the active site where the G cofactor binds was basically looking at that 3' hydroxyl group, which would normally have an oxygen in its place as the atom, and seeing whether when the G cofactor comes in, if we substituted that oxygen atom to a sulfur, which is depicted here, any difference was observed in the catalytic reaction. And the question becomes, well, why did they mutate the oxygen, as we saw on the previous slide, that is required for where the nucleophilic attacks occurs, to a sulfur? The interesting thing about sulfur is that it will not coordinate to magnesium. Okay, so if magnesium is required in the active site and the coordination to the oxygen is important for catalysis, if we substitute that atom to an atom that does not coordinate to magnesium, then essentially we should be able to observe no catalytic reaction occurring. Okay, so we know that these catalytic reactions require or are probably stimulated by divalent metal ions, but really it's difficult to separate the contribution of these metal ions to the formation of the tertiary structure from a more direct role in catalysis. So again, these experiments help address this question, and we have a blotch over here which we're going to walk through. So basically a synthetic substrate was generated. So we can call this our synthetic substrate. Okay, in which the 3' oxygen atom at the cleavage site, so for our cofactor was to come in, this is where the nucleophilic attack would happen at that particular atom, is replaced by a sulfur. And then we look at the generation of smaller fragment or cleavage of the product. So this is our uncleaved product here at the top on this gel versus our cleaved product, which is much smaller at the bottom of the gel. Okay, and this was actually carried out, and again, using two different metal ions, so magnesium and manganese, and two different atoms. So either in this position we had an oxygen or a sulfur group. And finally, these numbers here, 0 to 26, just represents the hours after treatment. So at 0 hours and then 26 hours later. So what the data actually reveals is that in the presence of magnesium, so we're focused on this set of results here, when the atom is an oxygen in that 3' position in our synthetic substrate, after 26 hours, we start to see cleavage, self-splicing of the intron once we provide that particular G cofactor. Okay? When the atom is changed to a sulfur, we can see that the cleavage to the smaller fragments does not occur after 26 hours. And this essentially establishes that you need magnesium to carry out the catalytic reaction in the active site, specifically because the sulfur atom does not coordinate to magnesium. Okay, and then as a positive control, they used manganese as the second set of experiments, because manganese will actually bind to both sulfur and oxygen. Okay, and you can see that in the presence of manganese, both of these synthetic substrates were cleaved after 26 hours, and actually see a much stronger complete cleavage in the presence of the manganese atom, with the manganese ion in the presence of the oxygen atom as well. Okay, so these data essentially reveal that that coordinating with the metal ions in the active site in the presence of the G cofactor is essential for catalysis to occur. And we see that in the presence of magnesium, when it's coordinated to oxygen, we get catalysis, but in the presence of a sulfur atom, we do not get catalysis. And this is because magnesium does not coordinate to sulfur. However, as a positive control, they actually also discovered that the manganese, which coordinates both to sulfur and oxygen, allowed for catalysis to occur. Okay, so these results, in summary, do indicate that the metal ions contribute directly to catalysis by coordinating to that 3' oxygen atom in the transition state, and this presumably stabilizes the developing negative charge on the leaving group when the first transesterification reaction occurs. Okay, so that basically summarizes the splicing lecture. And again, just a reminder, we have the group 1 and the group 2 self-splicing introns that were discovered. And in both cases, if we remember, for the group 1 and for the group 2, we have, again, the joining of exon 1 and exon 2 together through 2 transesterification reactions, much similar to what we saw with the external spliceosome. This can often be referred to as an external catalyst, and it's more easily regulated, more easily and readily regulated. Okay. And we can see the major transition, if we go across from group 1 to group 2 to the spliceosome, was basically the transfer of catalytic power from the intron itself. So from here to other molecules, so assembly of the SNRPs to make the spliceosome. And this resulted in the fact that introns then were no longer constrained to provide the catalytic center for the spliceosome. So maybe some evolutionary benefits to having this external catalyst carry out splicing mechanisms as compared to the introns themselves. Okay, so just a summary. In modern higher eukaryotes, the spliceosome is responsible for most RNA splicing. In other systems, they have self-splicing RNA introns. They are catalytic. They depend on magnesium metal ions for not only the 3D structure, but also the active site. The group 1s will use the guanosine cofactor as the starting point for the first transesterification reaction. Remember, it's not as an energy source, but rather to provide that 3' hydroxyl nucleophilic attack. And again, through the work of Czech and Altman, it was the first evidence that RNA is basically a biological catalyst. And then finally, if you want to research yourselves, you can actually look at how many viruses have ribozymes that can cleave RNA genomes. And again, they will use different methods of catalysis using either metals or base functionalities to assist with this process. So that concludes the splicing lecture. And now we'll focus on translation and how we can actually use all of these transcripts, the rRNA, the mRNA, and the tRNA, to help us synthesize proteins.